week and how, let's try to remember. Like specifically, like it helps us like kind of like target specific sections of the membrane.
Mm-hmm. And what else we talked about? What else we talked about?
General introduction to some kinds of lipids like fatty acids,
Mm-hmm.
triglycerides, and sphingolipids, that sort of thing.
Mm-hmm. Then what else?
Learned about like the difference between like primary and secondary active transport. Primary is like just like the simple one's kind of just against the gradient.
Mm-hmm.
The secondary one's like more complex.
Mm-hmm.
It's like a combination of like the primary one, but there's also like another transport involved, which kind of like transports like a second substrate into this cell. I mean membrane.
Mm-hmm. And what else we talked about? What else, guys? Anything rings a bell?
We spoke a bit about like omega-3 and vitamin D synthesis,
Okay.
and
what else
how
we
like
talked about?
fish contain like natural DHA.
Yeah, that's good. So what else we talked about?
We talked about the lipids that make up waxes and like bird feathers and honeycomb.
Yeah. And okay, good. Okay, so did we stop here?
Yeah.
Okay, so let's talk about this slide a little bit. So just to brush up the memory. So what we'll talk about today is about the polar structural lipids. So basically, this polar structural lipids contains the head groups is a polar group, and the tails are non-polar group. Now you also have we know that the head groups usually depends on the surface properties of the membrane. Okay, so we also know that different organisms have got different lipid head group compositions, which we kind of touched upon briefly in the previous lectures, and different tissues also do have different lipid head group compositions. Okay, so in terms of glycerophospholipids, we'll talk about it briefly here. So these are the primary constituents of cell membranes. Okay, so they do have this first and the second hydroxyl groups, or the yellow glycerol 3 phosphate. Okay, and the head groups are usually charged based on the physiological pH. Okay, so now these are some of the examples of glycerophospholipids. So for example, phosphorylserine, phosphorylcholine, phosphatidylcholine, phosphorylglycerol, cardiolipin. These are various glycerophospholipids. Okay, so you don't have to memorize any of this formula. Just just remember some of the examples of the glycerophospholipids. Okay, so and usually these all are dependent on the pH. Okay, so you can see that net charge of the pH varies. You can see for each of these glycerophospholipids. Okay, so most of them are acidic. Okay, and a couple of them are, well, most of them are acidic pH. Okay, so phosphorylcholine is a major component of the eukaryotic cell membrane. So many prokaryotes, including E. coli, cannot synthesize these lipids. So membranes usually, the prokaryotes usually do not contain any of this phosphorylcholine. Okay, just keep that in mind. But the major component of the eukaryotic cell membranes are the phosphorylcholine. Okay, so all right, so now in terms of the ether lipids, these are otherwise called as plasmalogen. So these are analogs of phosphorylethylamine. Okay, so they are usually common in vertebrate heart tissues. Mostly it's prevalent, actually, I would say, in the heart tissues. And they are found in some protozoa and anaerobic bacteria. Most of this function of this particular ether lipids are not very well understood. For example, how does this ether lipids increase membrane rigidity? What are the sources of signaling lipids, and what type of antioxidants, are they antioxidants? We do not know anything about it. And, so some of them are being cleaved by lipases, so a lot of unknown, questions are there, and which I think, needs further investigations. But the most important thing is nobody knows about its function, in terms of how they increase membrane rigidity and so on. Okay, so it's still less well studied lipid molecule. Okay. So in terms of ether lipids, one example I can give you is the platelets activating factor. Okay, so these platelets activating factors are important. So keep in mind, okay, these are very important plasminogens, which are the glycerophospholipids. Okay, they are very important for inflammation. Okay, especially they stimulate the aggregation of the blood platelets. Okay, so most important thing is, this is the first signaling lipid to be identified, but main important factor is inflammation. Okay, so and this is some of the structures here where you can see the head group in the ethanolamine is in the plasma. So the head group is the ethanolamine in the plasminogens, and the choline is the platelet activating factor. Okay, so and these are very important for inflammation. Okay, so I mean, this lecture is going to be like this. Okay, so it's mostly basics about lipids. Okay, so it's nothing more concrete, but I think as we go through the lectures, you'll find something interesting coming up, in terms of crystallography and stuff like that, and it'll be very interesting. But for the first few slides, it's going to be like all basic information. Okay, so sphingolipids. Okay, the sphingolipids are the backbone. Is not the glycerol, because these are not glycerol phospholipids. Okay, so the backbone of the sphingolipids are the long chain amino alcohol called sphingosine. Okay, and the fatty acids is joined to a sphingosine via an amide linkage, rather than an ester linkage. Okay, these are usually seen in the lipids. So the polar head group here is a sphingosine. Okay, and these are connected by a glycosidic or a phosphodiester linkage. They're mostly these sphingolipids are mostly found in the outer face of the plasma membranes. Okay, they are very, very predominant. Okay, so here are some of the examples of the sphingolipids. Okay, you have sphingomyelin, ceramide, and globoside and ganglioside. So there's a lot of complex. These are very complex sphingolipids. Okay, and by the way, keep in mind these are not dependent on the pH. Okay, so and some of the standard ways of how you represent this, they are shown there, but it's not really relevant here in this context. Okay, so but just remember some of the examples of the sphingolipids. Okay, so. Okay, so sphingomyelin, again, this is sphingomyelin is very abundant in the myelin sheath, okay, that surrounds some of the nerve cells in the animals. And the sphingosine plus any amide-linked fatty acids forms a ceramide. These are usually attached to the alcoholic group called the phosphocholine. Okay, so those phosphocholine are the ones that attach the alcohol, but the ceramides are usually a combinations of sphingosine and the amide-linked fatty acids. Okay, so and usually the phosphorylcholine and the sphingomyelin. So the sphingomyelins are the sphingolipids, and the phosphorylcholine are the glycerophospholipids. They do have similar dimensions and physical properties. Okay, but they have different roles in the membranes. Okay, so so this is one example where we're talking about sphingomyelin is structurally similar to the phosphorylcholine. So here is one example to show, protein, just two molecules structurally similar, but yet they have a different function. Okay, so just because they are similar in structure, it doesn't mean they have similar function. Okay, so this goes back again to the prediction of function, where we talked about, sometimes we can assign function based on the structural similarity. Yeah, We talked about that, yeah. So the point here is that yes, it is true that you can assign functions based on structural similarity, but again, these are putative or hypothetical function. So it has to be confirmed, because sometimes two proteins can have a similar structure, yet they can have different function. Okay, just keep that in mind. Okay, so a guild by association is possible, but still experimentally it has to be verified. Okay, so a structural proteins with similar structure can still have different functions. Okay, but the point about prediction is actually that you can assign function based on structural similarity, whether it's the same function or different function. Okay, it doesn't really matter, but at least you can assign function based on guild by association principle. Okay, all right, so here's one example to show the glycosphingolipids in the blood groups. So usually our blood groups, you know, that are determined by the type of sugar molecules that are located on the head groups, okay, in the glycosphingolipids. Okay, so for example, the structure of the sugar is determined by the expression of the glycosyl transferases, the enzymes. Okay, so for example, if you have individuals with no active glycosyl transferase, then they usually do have the O antigen. Okay, so if a person is having an A blood group, that means they do have glycosyl transferases and N-acetyl glycosylamine. Sorry, N-acetyl galactosamine. Okay, so whereas in the case of a B blood group, okay, so individuals will have a glycosyl transferase that transfers the galactose group. Okay, so this is how you differentiate these blood groups based on the glycosphingolipids. Okay, so we don't have to worry about the slide. It's just basically the summarizing what I told here. It's just a way of representing things, how the O antigen, A antigen, and B antigens are actually arranged. Okay, so this each one of them is showing the head groups, you know, of those glycosphingolipids. But the most important thing is these 3 points. Okay, so just this is a very key important point. So basically, how do you define an individual with an O antigen or A blood group or the B blood group? So a person who is having no active glycosyl transferase will have the O antigen, and the A blood group means the individual will have a glycosyl transferase, you know, the transfers and N-acetyl galactosamine group. And whereas in the case of B blood group, you have a glycosyl transferase that transfers the galactose group. So these 3 important features determines the O antigen, A blood group, and B blood group. Okay, so here's one example to show again, you know, how a defect in the lipid molecules can lead to a number of diseases. Okay, so for example, you have what is this called? So each one of them. So you have this each one of these molecules. You have different head groups, how they are arranged. You know, don't worry about that. Each one of them represents each one of this. So this is a glucosamine, galactosamine, you know, and N-acetylamine, you know, all those things. But I think the most important point here is that a mutation or a defect in these lipid molecules can lead to various disorders, like, for example, generalized gangliosidosis and Tay-Sachs disease, you know, Gaucher's disease, Niemann-Pick's disease. This is a very, very important one. Basically, what it is showing is actually, if you have a mutation in this in the sphingomyelinase, you know, so that means, like, you know, that can lead to your Niemann-Pick disease. This is actually very well still a lot of people in Atlantic province, you know, they are suffering with this Niemann-Pick disease. Okay, and Fabry disease at some point of time in Saskatchewan also, you know, people were suffering with the Fabry disease. Okay, it's basically a mutation in the galactosidase. Okay, so that's what it means. Okay, so and so basically the point here, what they're trying to say is, you know, that each one of this defect in some of these enzymes, you know, can lead to enzymes in the lipid pathway can lead to some of these diseases. Okay, so and okay, so sterols and cholesterol. So, you know, sterols, they have the steroid nucleus is almost planar, and, you know, and this is a chemical structure showing the steroid nucleus. The carbon atoms are all numbered in the, you know, So basically, this is just a steroid nucleus, and its derivatives are being shown here. Okay, so and each of these carbon moieties are shown how they are arranged in the polar head group. Okay, so I mean, again, this is nothing. You don't have to memorize the structure, but just basically the one take-home message here is that that the cholesterol, the chemical structure of this cholesterol, they do have a steroid nucleus. Okay, so and here this is very important. Here are some of the physiological roles of sterols. Okay, so sterols do have, they modulate fluidity and permeability. That's very important. Most of the bacteria lack sterols. So what do they have then if they lack sterols? What do they have instead of, so bacteria lack sterols, what do they have? We talked about it before. What do they have? Bacteria that lack sterols.
Peptidoglycan.
What's that?
Peptidoglycan.
No. So they have hapenols. Okay. So hapenols. Okay. So bacteria do not have sterols. They have hapenols instead of sterols. Okay. So and now most of these mammals, like us, humans, obtain cholesterol from food or synthesize, de novo in the liver. Okay, now usually the cholesterols are low density lipoproteins. They're not good for health because they clog arteries. Okay, so and many hormones are derivatives of sterols. Okay, so very important thing, the bacteria that lack sterols, they do have hapenols, okay, instead of sterols, but eukaryotic cells do have the cholesterol and the sterols. Okay, keep that in mind. Okay, so physiological roles of sterols for their functions. So mostly they interact with the proteins in the plasma membrane, not only for the regulation and transport of sterols through the cell, but also the context of cell receptor systems. Okay, so we do have a lipid sterol or lipid protein interaction. So usually disruption of this interaction, can cause a lot of issues in terms of the disruption of this lipid sterol protein interactions can lead to disruptions in the function as well as in terms of the context of diseases as well. Okay, so and these disruptions are quite frequent in the neuronal tissues, but again, this disruption can happen for any particular reason. Maybe the physiological, the physiology, if the physiology changes because of the environmental cue or because of the mutation, then these type of disruptions can happen. Okay, so here are some of the examples here where these are some of the genes that are involved in various processes of the sterol. Okay, so what is being shown here is that like the dark blue color here represents some of the genes that are being previously described, and there are some of the there are a lot of discoveries going on. They continue to discover a lot of new functions for the genes. So some of this, so there are about 50 genes out there that are newly discovered actually. Okay, and these are in various processes like glycerolphospholipid metabolism, the snare, and they are involved in synaptic vesicles, membrane signaling, and glycan synthesis, so on and so forth. So there are a lot of new functions being constantly discovered. Okay, you have to keep in mind, anybody knows anything about this GO categories? So what does it mean GO categories mean here? What does GO categories mean? Any idea?
Gene orthology.
Gene orthology, no, but gene is correct. G is for gene. O is for ontology. Okay, what does it mean, gene ontology? What does it mean, gene ontology? What does it mean, gene ontology categories mean? So usually what happens is, in a molecular biology, that in order for us to assign function to a specific category, like, for example, there's always a parental hierarchy, and then the child hierarchy, and so on and so forth. So basically, how can you classify a protein to a specific class of functional groups? Okay, so do you guys know about NCBI? Yeah. Okay.
Yes.
So let's take an example. Let's go here and let's just look at it. Okay. So all right. So you see the Go categories here? Yeah. So this is gene ontology. Okay. So basically, gene ontology has got 3 categories. Okay. It's called biological process, molecular function, and cellular component. Okay. So basically, what does it mean? This is what I mean. Okay. So you see here, look, for example, I'll take a look at this. Okay. Can you see my screen here? Can you see my screen here? Yeah. So you see, metabolic process is the parental category. Okay. So this is a hierarchy. Okay. And you still have child hierarchy, and so and so on and so forth. You can just see the hierarchy is being branched out into small subgroups. Am I right? So for example, if you want to assign a gene to a hexose biosynthetic process, then you can actually go back and trace this pathway and see the major pathways, the metabolic process. So you can basically go back and backtrack them. So okay, so this is how. So if you know a function of the protein, and you can assign the functional grouping, which group it belongs to. Do you know what I mean? So for example, if you look at this, for example, just for example, okay, so if you want to divide these genes into various processes like signaling, protein transport, and carbon metabolism, this all are coming based on this hierarchy of the gene ontology. You understand? So this is how you actually group the genes into a specific functional categories. Okay. Do you follow? Yeah. So that's how you actually classify these genes into specific groups. Okay. So and that's what this means. Okay. So the 50 genes have been grouped into various functional categories. Okay. So but the most important thing is it has got molecular function, cellular, biological function, molecular function, and cellular component. Okay, so each one of them, biological function is based on the biological processes, molecular function is based on molecular processes, cellular component is based on subcellular localization. So where does it localize to? So that's how it's been. So each gene, if you go, you can actually figure out, where do they actually, we can classify them to. For example, for unknown genes, the gene of unknown function, nothing you can do about it, and those will not have any group in the Go categories. Do you follow? Yeah, because those are a gene of unknown function, so they do not have any categories. Okay, so but only those that are very well categorized, very well characterized, are the only ones. Those will be assigned to a specific group. Okay. All right. So. And here are the sterols in the nervous system. So for example, let's take a step back, so why mouse models are very important. Why mouse models are very important.
You're allowed to test on them ethically.
Okay, so give me some examples. That's good. That's a good point. So it allows them to test them, but so what are we testing them for?
You can test drug application prior to human trials.
Okay, such as?
Yeah, I don't know an example, but say you have what is the word? I don't know, some sort of cancer treatment, something you want to try it out on humans, right. But you have to go through the process of testing it on something else first.
All right, cancer, that's good. That's a good point. So for cancer, we can actually induce this cancer cells into the mouse and look at the tumors and treat them with the drug and see what the tumor efficacy looks like, or the drug efficacy looks on the tumor in the mouse. Yeah. Okay, so that's one way to study them. But can you study psychiatric disorders in mouse?
Rian said in the chat, mostly not. I think I agree.
Okay, so what about neurodegeneration?
That you could do.
You can do. So why not psychiatric disorder?
It's hard to categorize what disorder they have if you can't communicate with it.
Okay, but still you can measure the behavioral studies can be conducted based on because psychiatric disorder is also a behavioral problem. Yeah, is that correct? So psychiatric disorder is a behavioral problem to some degree. Am I right? So because they'll have mania, they'll have seizures, and they have different episodes they have. So these are recurrent episodes. Am I right? So you can study to some degree some sort of a behavioral problems in the mouse. Okay, but definitely psychiatric disorder is the biggest problem in the mouse because and you cannot study them completely. Okay, so my point is mouse models are good for some disease type, but not for, it's not a one-fit-all approach kind of a thing, that mouse models are good for everything. So mouse models are good for neurodegeneration. For example, you can measure tremor, you can measure neurodegeneration in the brain, in the mouse, in the brain regions. Those are all you can actually do. We can study them using mouse. Okay, so mouse models are good for some things, but not for everything. Okay, so how do you overcome it? How do you overcome? So I told you that mouse models are good for something, but not for everything. So for example, if I want to study something about the psychiatric disorder, and we talked about mouse models are not good ones, so how do I overcome with it? So what do I do to study them? So you understand, gene function can be associated with the pathology of the disease phenotype. So usually the good way to measure the phenotype is through mouse models. For example, if a person is suffering from a pancreatic cancer or a breast cancer or prostate cancer or whatever it is, okay, and you can replicate those ones ex vivo or in vivo through the mouse models, by replicating the tumors in those mouse models, and then we can assess them or what the efficacy of those treatment on those tumors. Okay, so it's a drug or whatever it is, you can study the efficacy of those drugs on the tumors. Okay, so but what happens in some cases where we cannot study them, and there should be a way to study them, right? So my question to you guys is actually, what are the ways that you can study the pathology of the disease in the event of or in the event where there is no mouse models are available to study? Think about it. What do you guys think? Doesn't matter right or wrong, okay? Just talk.
Can we replicate those phenotypes in cells, like in vivo, instead of like a mouse?
Sure. How do you do that? That's why my question is. In what type of cells you want to replicate it?
Tumor cells?
Tumor cells. So why would you say tumor cells? So for example, okay, well, what do you mean by tumor cells?
Wouldn't that replicate? Because they're always going to be replicating.
Okay, but replication is not how you measure the phenotype, am I right? It's a disease pathology has to be expressed. For example, if I want to study a person with a Parkinson's disease, is the tumor cells are going to be helpful?
Oh, no.
Yeah. Okay, so how would you do that? Think about it. It's okay. It's taking time. It's fine. Just think about it. Yes, Brian. Brian, did you raise your hand or what?
She has answers in the chat.
Answers in the chat. Okay, all right. Okay. Okay, what? Okay, you can explain. You can explain. You're talking right to me. Okay, so. Okay, so what is the point there? Are you there, Brian? Okay, well, I don't understand why he cannot talk. Okay, so one way we can do is, so what does the chat message says, guys?
Mentions like the gene knockouts or downs that can be done in mice. Depends.
Gene knockouts. Okay, so well, gene knockout in mice is, well, I don't. My point was I can't. Well, my point was I don't even want to do the mice. Am I right? I want to do something outside of the mice because to replicate the findings. So my question is, how do we do that outside of the mice? So what we can do is what we call them as an induced pluripotent stem cells. Have you guys heard about the induced pluripotent stem cells?
In the chat, it looks like Brianne has.
What?
She's mentioning stem cells. Yeah,
okay, well, stem cells is a general term, okay? So not all stem cells are induced pluripotent stem cells, okay? So, so how many of you guys know about induced pluripotent stem cells? So how do you do that? Okay, so basically what you can do is you can actually take a human sample, whether it's a healthy or preferably, I mean, if you want to study the pathology of the disease, then in the disease patients, what you can do is you can isolate a fibroblast just from the keratinocyte, from the skin fibroblast, or whole blood cells, or from lymphoma. You can just take those ones and then you can convert those into a fibroblast. Okay, so those fibroblasts are a mass of tissues. Okay, and those fibroblasts, upon adding growth factors and all those things, you can convert them into fibroblasts into the induced pluripotent stem cells. Now, and these induced pluripotent stem cells, it's a stem cell. Now you can convert them into a specific neurons. Like for example, if you want to study more about the dopamine neurons, you can convert the iPSCs into dopamine neurons, or if you want to convert them into motor neurons or cortical neurons. So whatever neuron type you want to do it, you can do it. Okay, so that's the beauty of it, because you can actually study now the pathology of the disease. Now you can do a knockout and all those things. You can do it it's extremely tedious work because it takes several months to generate this, not years, but just several months to generate this iPSC. iPSCs, and then from iPSCs to the neurons. Okay, so and generally using growth factors, you can convert them into a specific neurons that you want to study in a disease patients directly. Okay, so this is one way to study the pathological symptoms. But again, you have to keep in mind. Okay, now you might argue, okay, how can I study the behavior of those of those symptoms. Am I right? I mean, that's unfortunately we cannot do this in the iPSCs, because all we can study about is actually the phenotype. You can measure them, but not like you can measure, like, you know, how much of neuron, how many neurons are dying, how many neurons are surviving, okay, in a disease patients, and do they have more tumors or less tumors, you know, all those things, you know, you can study them using well, not the tumors per se, but you can measure the cancer proliferation cells, you know, all those things you can study. So cancer proliferation, or dopamine neurons, or cortical neurons, or motor neurons, hippocampal neurons, or whatever it is, different types of neurons, you can convert them and you can study. So for example, if you want to study for example, if you want to study the function of this particular protein called STAR, you know, and then you can make a knockout in the neurons and study in a specific neuron type, you know, what does this particular gene does, you know, or doing, you know, in this particular neuron cells. But what is the disadvantage is actually, you know, there's always a heterogeneity is there, am I right? Because patients to patients, you know, and all the iPSCs also, you know, may not reflect the functional complexity or the structural complexity of the brain regions. Okay, so it can give you, the induced pluripotent stem cells will give you about the neurons, okay, what type of neurons are there in the BD patients, or sorry, in the disease patients, and study them, okay, in the specific neurons. But it doesn't give the structural and the functional complexity, or because these are very complex, brain regions are very complex, okay. So in order for you to study more in a specific, because, you know, once you generate these neurons, okay, so from the brain regions, for example, dopamine or motor neurons, it's always not 100% pure neuronal population, because it's a mixture of glial cells and the neurons. Okay, so there's no pure clonal and neuronal selection. Okay, so in order for you to do that, you can always go back. Okay, so now I'll ask you another question. So even if you isolate iPSCs, okay, and then convert them into neurons, does that replicate the entire 3D structure of the brain regions, or not the 3D structure, the functional integrity or the functional characteristics features of the brain region completely, or not?
I'm guessing no. You're probably just getting a mass of cells, right, or a mass of neurons.
Yeah. Okay. So does that imitate, or does that replicate the exact brain brain features, or not? The characteristic features, or not?
Probably not even close.
Yeah. So because of the mixture of cells and heterogeneity, you know, it's always a problem. So that's why people are going to go one step further, and what we call them as called organoids. Okay, so those are called brain organoids. Okay, and these brain organoids, you can basically grow the brain in a dish, basically. Okay, so that's what it means. Okay, so you can actually now the cortical organoids, okay, the dopamine organoids, all these things, even you can create a triple negative breast cancer organoids. Okay, so basically tumor organoids. So basically you can create whatever you want. Okay, and these are called a 3D structural organoids. Okay, so that retains the functional capacity of the brain regions. Okay, everything is intact. Now you can actually study and mimic, you know, the pathological situations. Okay, if there's not you know, if there's no other way to study things in the mouse models, you can always do this in an iPSCs or in a brain organoids. Okay, so just to keep in mind. Okay, this is one way to study the pathological features of any disease type, you know, if there is an absence or lack of mouse models. Okay, but of course the disadvantages in both this iPSCs and brain organoids is that, you know, we cannot study the behavioral patterns of those. So we cannot study the behavioral symptoms. For example, the psychiatric symptoms. No, we cannot study them. So we need, for behavioral studies, we can use, like, you know, mouse, for example. You know, you can knock out a gene, you know, in a mouse and see, you know, what the behavioral changes are. For example, how fast they can run, there are a lot of assays out there. Okay, so we don't want to go through these assays, but it's called like maze assay. Basically, like you just put them in a jar and allow the mouse to figure out how it can come out, so how much time it takes. So all those things are very behavioral things, so that I think we can assess the psychiatric problems in a mouse, through behavioral studies. Okay, but that we cannot do this in an iPSCs or in the brain organoids. Okay, so just for your own information about this topic, okay, I want to say, not mouse models are always there, but but there are other ways you can also study the pathology and the pathology of the disease. Okay, so now here we explained about like some of these genes and their function. We talked about both categories. We talked about some of these new genes are being discovered. Okay, so and here is here is an example to show how the mouse models, transgenic mouse models, can be used. Okay, so basically, so you can see that there's a gene name is listed here. You see FASN and DHCR7 and DHCR24, SQS. These all are gene names. Okay, so mutation in this genes can lead to various impairment in the lipid synthesis or the myelin synthesis or the cholesterol trafficking or impairment in the cholesterol derivatives. Do you follow? Okay, so there are a lot of disease types out there. Okay, when you impair some of these genes. Okay. All right. So now, okay. So steroid hormones. Okay, so steroids are oxidized derivatives of sterols, and the steroids do have sterol nucleus, and mostly they are polar. Okay, and then the steroid hormones are synthesized from cholesterol in gonads and adrenal glands, and mostly they are carried through the body in the bloodstream and usually attached to the carrier proteins. And again, so the steroid hormones are male and female sex hormones. Okay, many of them are. Okay, so now to touch upon this aspect of the male and female. Okay, so now you have to keep in mind. Okay, so how many of you guys know about so let's say in your research when you do some experimental experiments. Okay, so whether it is a human, okay, or well, mostly in human or mouse, or Drosophila or any other model systems you study, what do you guys think about male and female subjects? How many of you guys think that it's very important to study male and female to include a male and female in your research design? Is it necessary?
Yes.
Why? Is it Patricia? No. Who is this?
It's
Yeah. Who?
Luke.
Okay, hello. Yeah. Why?
Because, well, men have a 24-hour hormone cycle and women have a month.
Forget about the hormones. I mean, like, we don't care about the hormones. I mean, I'm just talking about in general. I mean, this is one example, but I'm just talking about in terms of research design. How many of you guys favor that we should consider both male and female subjects in your research design? So, for example, if I study, let's say, if I want to study human cell lines for that matter, okay, so does it really matter? It has to be from male and female subjects, okay? So, or any research design I do it, why I should consider male and female subjects, or why I should not consider male and female subjects. So what do you got? What is your perspective on the area of of including male and female subjects in your research design?
Honestly, off the top of my head, I do it just to see if there was a difference in my result. So as a control, just to check.
What type of control are you looking for, Luke?
It depends on the specific thing, but what I'm saying is just to see if there is a difference.
Let's say breast cancer. Okay, let's say breast cancer. Okay, so I want to study the efficacy of the drug on a breast cancer. Okay, so, and I want to do this in a mouse. Okay, all right.
Mhm.
So, what do you think about the male and female subjects here?
I'm so used to hearing about women having breast cancer, so
Okay.
I don't even know if men have
Well-
breast cancer.
So you think men do not have a breast cancer?
Well, they have breast tissue, I guess,
Yeah,
so they could have breast cancer.
yeah, for sure they do have breast cancer, but it's not as prevalent as in the female. But the point here is that my question here is that should I include a male and female subjects? When I say subjects, it can be anything. It can be animal, or it can be individual, us, or Drosophila flies or whatever. Should I include that male and female subjects into my research design or not?
Yes, because what if your treatment doesn't work for men, but it worked for women?
Okay, so which means you'll include them.
Yes.
Okay, so and okay, so what about psychiatric disorder, let's say?
Oh, absolutely, yes. Especially not even in like a biological sense, but like socially, women and men are like socialized differently, so that can affect your.
Okay, so what about in Drosophila? Let's say take a fly, Drosophila. Okay, so I want to study I want to study neurodegeneration in Drosophila. Okay, so what about that?
In a
Could I
fly?
Yeah, I mean, what do you think in Drosophila? Do you know that the pink one Parkin example I just showed you about before, about pink one Parkin in the neurodegeneration, that came actually from the Drosophila flies?
Wow.
Okay, yeah, do you know that?
No.
Yeah, Drosophila is a good model system. Okay, so many. Okay, I want to touch upon here. Okay, model systems are pretty good because we cannot replicate them. But right now we have all this iPSCs and brain organoids and everything that makes it a lot more sophisticated to study things. But again, still much more expensive. Not all labs can afford to do that. But if you can study yeast, you can use yeast. Do you know that people have studied neurodegeneration in yeast? Okay, so they have studied it. Okay, but it's not like actually you cannot replicate this. This is symptoms, but the biological functions, yes, they have studied it. But the pathological symptom of a disease phenotype, they have studied it in Drosophila. Okay, what about C. elegans, a worm? That's a very good model system. Okay, all right, so people have studied that. Okay, so that's a really, because these are the type of things where you can easily study the gene knockouts. Okay, you can knock out the gene very easily because it's very small structures, much easier to handle it. So people have studied that. Okay, and bacteria, E. coli is a very good model system, but not in the context of human pathology. Okay, but I'm just talking about for the infectious diseases. Okay, so and again, mouse is another good model system. Okay, so these all are beside the point of iPSCs and brain organoids. My question to you guys is, I think the reason why I'm talking about this in a great length is because as we move into this area of new generation, and I think it's extremely important that we consider this male and female subjects in all research designs. Okay, my question to you guys is why it is so important.
I'm not sure if this is like of any importance at all, but like males have like the Y chromosomes, but females don't.
Yeah, I mean, like, regardless of the Y chromosome, the X chromosome doesn't really matter. Okay, my point is, so why do we even bother about studying them in the first place? Why do we even need to include them in the first place in every single research design?
Because they're different.
Okay, so just because they're different doesn't mean it's important.
Just
So we can.
enriches,
So why only male and female? Why not transgender?
you should include that in your research design as well. Yes, but
Okay, so why? That's why my question is why.
because you're covering more ground, right? The
More
more
ground for what?
subjects you include, the more data points you get.
Okay,
You might
that's
get more
fair.
variance with different genders.
those all are fair points. Okay, why?
It's more representative of the population.
Okay, why? Okay, these all are great points. Okay, you guys are telling me adding all these things are representation and all the things are great, but why? Why do, okay, all right. You have only $10,000 research funding, okay? All right. Now, you are supposed to do a function of a gene, okay? Let's say psychiatric disorder, okay? You don't have funding to recruit all these populations to study your gene function. So my question is, you still have to include them. My question is why?
Because you're still releasing it to the population and not just men or just women. So
what population, Luke?
If you're like, sorry, I forget the example, but if it's a drug trial and you only have, for example, men in your sample, the drug isn't just going to be prescribed to men.
That's good.
So you
need what?
You need to include women or transgender people and everybody.
Exactly. So that's good point. So now, because it all boils down, this is why Canadian government and the National Institute of Health Research and the Canadian Institute of Health Research, they are pushing forward to have this sex and gender in all your samples. Okay, so now whether, for example, if you are studying about HEC293 cells, HEC293 cells is a human embryonic kidney cells. Am I right? Yeah. So these are regular human cell lines. Okay. These cell lines are coming from women. Okay. All right. So, but now they want to know even if you want to test them, you need to make sure that whether it's a cell line or whether it's a brain organoids or whether it's iPSCs or whether even it's a mouse, doesn't matter. Even if it's a mouse, they should. Mouse do not have a transgender. Okay, but I think so even if it's a mouse, it's a male and female. Okay, so my point is there should be an equal representation of different genders and sex. We should evaluate them. Why it is very important? Because as we go down the road, there's a complications are there. What are the complications? So Alok said that for drug trial, it's very important to see how we prescribe. So just go a little bit more in detail. What are the disadvantages of that? So if you don't include them, or if you include them, let's say you include them, then what? Okay, so now, okay, let's take an example. Okay, so there is a psychiatric disorder. Okay, so I'm sorry to go by psychiatric disorder, but anyway, so let's take an example, lamotrigine. Okay, lamotrigine is a new drug in the market. Okay, and of course, you want to test this lamotrigine in the bipolar disorder, and now you have a female, male, and blah blah blah. Okay, so now you're evaluating them. Okay, well, you're coming up with the consensus like, okay, well, you know what, this drug worked so good in the female. This drug did not work very well in this male population. Okay, so what?
You can make a recommendation for its prescription then?
To whom?
To whoever's marketing it and to whatever doctor, physician, or psychiatrist is giving it out to patients.
Okay, so is that how the clinicians are actually doing it?
Sorry, what do you mean?
Okay, so you just said the data suggests that blah blah blah. Okay, so you got the data out. This many percentage of the females are positive with the positive treatment, and this many males are negative, blah blah blah. So you came up with a consensus. So now, who has to prescribe it?
The physicians or psychiatrists have to prescribe it, not you.
Okay, sure, that's right, fair enough. But the physician, is it what they are doing it?
Is it, sorry, what's that? Say that again.
Is a physician, how they are prescribing it? Are they prescribing X amount of dose to the female, X amount of dose to the male? Is it how they are prescribing, or are they just prescribing? You can take lamotrigine 1 or 2 doses per day.
Yeah, they're giving a certain dosage and timing or whatever.
Exactly. So still the physicians are not clear the impact of the doses, because this is what the study says. Because any drug that you prescribe, so starting from the clinical trial to the physician's office, okay, there is no interconnection between these people, because people who are marketing, they're marketing it. Okay, there is no evidence-based study to show that, like, you know, how a woman has to be prescribed and how a man has to be prescribed. Do you understand? So if a man and a woman goes to the treatment, the physician is going to say, you take lamotrigine 1 or 2 doses. Is he going to say, he or she is going to say, for female, you take a half a dose lamotrigine for 10 days. For male, take 1 full dose for 10 days. Is he going to say that?
He's only going to say it based on what the study's given him or whatever the marketing company has given him.
Yeah, exactly. Yeah, well, he or she, whoever the physician is, the physician is going to say, you know, so unless, I mean, in every single trial, the problem is, the problem here is that, I mean, the reason why I'm spending so much time on this topic is because it's extremely important. As a young generation, you know, when you guys go into medical field or medicine or any other profession, you know, you have to understand how the country is moving forward, in which direction the research is going forward. Sex and gender is absolutely critical, because you cannot prescribe, because there are a lot of hormonal changes that are happening in the female compared to men. You know what I mean? Any drugs will have a side effects, you know, especially for female, because of the hormonal imbalances happening. They have a lot more side effects compared to the male. Okay, so what they're trying to do is, for every drug that they are being prescribed, there should be an evidence-based research. Meaning, you know, most of the pharmaceutical companies do not, even though they have this test. Okay, let me ask you this question, okay? You have breast cancers are prevalent in where?
Women.
Okay. And in your clinical trial, are there going to be more women or more men?
More women.
Yeah. Is it fair?
Yes.
Why it is fair?
Because it's more prevalent in women.
That's the problem, Alok. That's the problem. Okay. So that's a problem, because that's what they want to defeat it. Okay. What they want to do is test the drug, because if you do this way, okay. So for example, psychiatric disorders are more prevalent in men age of 10 to 25. Okay. So now what happened to women? Am I right? So the point here is that there should be an equal representation of how you test a drug trial. Okay. And that's what the government is pushing forward. So, which means in every single study, there should be an equal representation, and this has to be an evidence-based research, and that would allow the physician, you know, to prescribe, you know, a dose for right. So instead of saying, you know, if you take Tylenol, let's say Tylenol, okay, so they're just going to say, take 2 Tylenols for adults, male or female, doesn't matter, take 2 Tylenols, am I right? Yeah. So, but those type of things has to be changed, which means, you know, how do you regulate these drugs to a woman? How do you regulate this drug to a transgender or to a male or whatever it is? Okay, and that's what they want to do it, because it has to be an evidence-based research. So that this is what it's called personalized medicine. Okay, a personalized medicine or a precision medicine. Okay, so which means each individual gets a proper recommendation of how you want to treat a person based on a personalized medicine. Okay, so just a concept. Okay, so anyway, let's keep moving forward. Okay, but I just threw in a lot of stuff for you guys, but this is what, you know, how research is going on. So every single time when you conduct a research, make sure that when you write an article, when you write a research grant or anything, it has to be a sex and gender has to be integrated into a research design. It's mandatory right now. Okay, all right. So here are some of the examples of steroid hormones. Okay, so basically, you know, testosterone, you know, estradiol, aldosterone, a lot of things. Okay, so a steroid, you know. So do you know? I mean, I every year I ask this question just to have, you know, see how many of you guys know about. You know, do you know what is this steroid coming from? This steroid, this growth regulator. Anybody?
I just searched this up. It comes from plants.
Yes, of course. That's what I'm asking you. So, what plant is famous in Saskatchewan?
Wheat.
Then
Canola.
Grasses.
canola. Okay. So, what about other one? Do you know brassica? What is brassica? Mustard. Okay, so that's what it is. Okay, so that's the name. It's called brassinosteroid. It's come from Brassica napus. Okay, all right. Okay, so now let's go back here. So biologically active lipids. Okay, so the biologically active lipids are usually present in a smaller amounts than storage or the structural lipids. Now they play a vital role in signaling molecules nearby cells. So most important active lipids are lipid soluble vitamins A, D, K. Okay, so those are the lipid soluble vitamins. So, and here's one other example to talk about is arachidonic acid derivatives of signaling lipids. So some of the examples like the thromboxanes, formation of blood clots, leukotrienes, those are important for smooth muscle contraction in lungs. Prostaglandins is important for inflammation and fever. So these are some of the examples of signaling lipids. Okay, so don't worry about all the structures and everything. Okay, so it's just so for example, the NSAIDs are nothing but these are non-steroidal anti-inflammatory drugs, okay, such as aspirin or ibuprofen and all those things. So they block the formation of the prostaglandin and thromboxanes, okay, so by inhibiting the enzyme called the cyclooxygenase. Okay, so but anyway, I mean that's not the point here. The point here is that just learn some examples of those signaling lipids. Okay, so now vitamin D regulates calcium uptake. So here we can talk a little bit more about. I can spend some time here. So basically, what is this called? The calciferol, the vitamin D3. Okay, so this is a vitamin D3. These are usually produced in the skin by UV radiation. Okay, so and this actually is being produced by this product coming from the 7-dehydrocholesterol. Okay, this dehydrocholesterol breaks the bond. Upon UV light, it breaks upon. The bond is being broken, and then it leads to this. So here's where the bond is being broken, and then that leads to this calciferol, the vitamin D3. Okay, the vitamin D3 then is added. The hydroxyl group is added to the carboxyl 25, the carbon chain 25 here, and that is actually in the kidney. Okay, so and then the second hydroxylation is happening. That's the C1, the carbon 1 chain here, and that is actually being produced here. Okay, and this is actually produced in the liver. Okay, so now this is actually these hormones, basically the dihydroxy vitamin D, it's called calcitriol, is actually regulates the metabolism of calcium in kidney, intestine, and bone. Okay, so to repeat again, the calciferol, the vitamin D3 is basically comes from this dihydrocholesterol. Upon UV light, it breaks down this bond here that leads to this calciferol, and which actually further in the carboxyl 25, it's actually a hydroxyl group is being added. That's happening in the kidney. And then sorry, the hydroxyl group is happening in the liver, and then in the kidney, it's actually the carbon 1 chain is happening. Okay, so this is in the liver, this is in the kidney. Okay, so C1. Okay, and that produces an active hormone called the calcitriol. This calcitriol regulates this metabolism of calcium in kidney, intestine, and bone. Okay, so this is how the vitamin D regulates the calcium uptake. Okay, so now vitamin A is very important because this is retinol is a derivative here. So they're usually involved in this visual pigmentation. Okay, so the hormones are involved in signaling. Okay, so what happens if you have this beta carotene? This is a precursor of the vitamin A. Okay, so now there is a cleavage in this beta carotene here that yields 2 molecules of vitamin A1 that's in the retinol. Okay, so this is where the cleavage happens, and then there's a vitamin A in the retinol. Okay, now this is in the panel A, and on the panel B, there's an oxidation is happening that converts the C15 here, the retinol to the aldehyde group. Okay, so here is the aldehyde group, and that leads to what you call them as a retinol here. Okay, and then further it produces what we call them as a retinoic acid here. Okay, that's upon oxidation of aldehyde to acid that forms retinoic acid here. Okay, so a hormone that's very important that regulates gene expression. Okay, so now what happens now? The retinol, this retinol, okay, combines with the protein called opsin, okay, to form rhodopsin. Okay, so the rhodopsin is a very, very important. Okay, so but it's not shown here. Okay, but they are usually produced in the nature. Okay, so rhodopsin. Okay, so now the rhodopsin molecules are usually visible in light. Okay, these are very important pigmentation. So you can just see. I can show you some snippets here. Okay, what does it say here? So they are usually visible in light. They undergo a series of photochemical reactions. Okay, so we talked about from here, there is a visual pigmentation that leads to retinoic acid. Now this retinoic acid are the hormonal signals that goes to the epithelial cell. Okay, now you also have this visible light. Okay, so basically what happens here is that when you have 11-cis retinol, upon visible light, it forms trans retinol. Okay, so basically, so the rhodopsin molecule is actually excited by a visible light. Okay, the cis retinol undergoes a series of photochemical reactions that convert into a all trans retinol. Okay, so and this leads to a change in the shape of the rhodopsin molecule. Okay, and then this retinol can be useful in the signaling to the brains and so on and so forth. Okay, so just to summarize it, so you have here a beta carotene that's a precursor of the vitamin A, and it undergoes a cleavage of this point of cleavage of the beta carotene that leads to 2 molecules. So one is a vitamin A1, okay, 2 molecules of vitamin A1, okay, and then upon oxidation, it converts the alcohol to aldehyde, okay, and that forms cis retinol. Okay, so and then you also have what it is called as retinoic acid also upon oxidation to aldehyde to acid. Okay, so one is alcohol to aldehyde, another one is aldehyde to acid. Okay, that's retinoic acid. Okay, the hormone that's important for gene expression. Okay, so the retinol then combines with a protein called opsin to form rhodopsin. Okay, so this rhodopsin molecule are usually, when they're excited by a visible light, okay, the 11-cis retinol undergoes a series of photochemical reactions that converts into all-trans retinol. Okay, so, and that leads to a change in the rhodopsin molecule structure. But anyway, the main functions of the retinoic acid is to send the signals to epithelial cells, and for all-trans retinol, it's involved in sending signals to the brain or the neuronal regions or to the brain regions, okay, via signal transduction. Okay, so do you guys follow? Okay. All right, so, and sorry, just a little bit smaller here. Okay, so here are some of the vitamin E, K, and other lipid quinones or antioxidants. Okay, so these are some of the antioxidants like vitamin E, you know, phytoquinones or the blood clotting cofactors, and warfarin is a blood anticoagulant, you know, and ubiquinone, a mitochondrial electron carrier, CoQ enzymes. Okay, and there's also plastoquinones. There's a chloroplast electron carrier. These are some of the biologically active isoprenoid compounds or the derivatives. Okay, so, and yeah, so these are very important in humans, you know, and bacterials, or in bacteria, the dolichols are very important for bacteria. But again, just remember some of these examples of the isoprenoid compounds. Okay, and the most some of them are the vitamin E and K and other lipids are antioxidants. Okay, so what else we have here? So these are some of the polyketides. These are biologically active lipids. Those are useful for medicinal purposes. Okay, so for example, lovastatin, erythromycin, and amphotericin. These are some of the examples of medicinal use. Do you know where we use this molecules called statins?
Is that for blood pressure?
Blood pressure or cholesterol?
Probably
so people,
cholesterol.
Yeah, when people are having cholesterols, you know, they usually recommend to lower the cholesterol level for lowering the cholesterols, they give the statin medicine. Okay, so this is prescribed for people who are having high high cholesterol levels. Okay, so let's take a break now. Okay, here we'll continue the lectures. Okay, so and let's meet around 7:30, guys. Okay, so let's take a break.